Sir, can I ask a question?
Yeah, please.
It's a very general question. I just wanted to know what do you think about the Nobel Prize in biology?
In biology.
Yeah.
So this year's biology Nobel went to
Charles
HCV,
Rice,
hepatitis C.
hepatitis C.
And also in chemistry as well, something
Yeah,
related to molecular biology.
yeah. So slowly you will see even the physics prize will also be given to molecular biologists in maybe a decade from now, because the biggest problems in this universe are now, which are yet to be solved, are in biology. Cell is, as we are moving in these molecular genetics lectures, you will see cell is still a black hole. We know the um, nuts and bolts of the cell, but we really don't know the mechanism. And I think hepatitis C Nobel, I mean, this is very well deserving, because if you read the history of hepatitis C, it's nearly 100-year-old problem. And it becomes even more important to discuss this because corona, in terms of, this is a viral era in which we are going through. All of us are suffering. And corona also jumps from, we still don't know its origin. They say it's coming from bats or animals. HCV also came from animals, and it was observed in vet clinics, in the veterinary, by the veterinarians, and in a horse breeding, what you call this, अंग्रेजी में क्या कहते हैं इसे यार, जहां आप लोग हॉर्सेस को ब्रीड करते हैं उसका एक खास नाम होता है. So there a veterinarian detected that horses are suffering from strange disease, and they're eventually their liver gets damaged, what now we call hepatocellular carcinoma in humans. So it took nearly, how much I would say, 60 to 70 years to discover that it has jumped from animals to humans. I think in late 70s or early 80s they started detecting this, because hepatitis A was already known, hepatitis B was known, E was known, I think, but they had no idea about this virus. And it was in 80s, I think, that Charles White, the other two fellows, they started working on this. But if you look at the history, I mean, they managed to clone this. They managed to prove in virology, we call this Koch postulate. Koch postulates means you clone an infectious clone, you clone virus or whatever bacteria, and then you reproduce those infections. You show that I cloned this particular DNA or RNA, and this molecule is causing actually the disease. So I think it's a very well deserving Nobel. It's even more important. But what wonders me when I look at Corona and these 20 years, I wonder that we have seen a bigger number of viruses jumping from animals to humans. If you go back earlier part of this century, so we had SARS, we had Ebola there, we had, I think there are 5 to 6 big jumps. Now, whether they are man-made, whether they are natural, I as a scientist don't believe natural evolution of viruses can be so fast that you detect it in animal and then it jumps. If hepatitis C was detected in 1920s or 30s in horses, and it was detected in humans in late 70s or so, or 60s, so it took 50, 60 years. And here we have very rapid jumps. So Charles also, I think if you want more detail, you should go to SSE. So Dr. Sabi sent an email to all of us. Charles White actually gave a talk in one of the symposium 2 years ago at LUMS. He couldn't be here physically, but he was very kind enough to give online talk, and it's a very good talk. If you want to read about more precise history and the transmission and how hepatitis C works, you should watch Charles that talk. It's a very well made. Whereas the other thing is CRISPR system, the bacterial immune system. There I have some reservations because my little knowledge tells me the person who discovered CRISPR system, which is the bacterial defense system, he is not included in the Nobel. And if I pronounce the names correctly, I think Jennifer or Jennifer Doana, and the other is Emanuela, I think. They actually used CRISPR-Cas system to develop tool to edit genomes. I think it could have been better if they included the one who discovered CRISPR-Cas. And if I recall correctly, it was Archaea. Archaea between bacteria and humans. It's in the evolutionary tree. So it was discovered in Archaea. So yes, it's a very interesting tool. Now you can design your babies yourself. You say there's a genetic disorder in my family, for example, deafness or whatever, and those disorders can be fixed, but one has to look at ethical issues before one goes on and use it in clinics. So 3 years ago, also chemistry Nobel went to phage display, the person who discovered how to do mutagenesis. She works on catalysis, and she discovered strategies about how to mutagenize, more or less every base pair in what you call in bacteria or phages. So she worked on phage display. As students, as faculty, what it actually highlights is that your education as undergrad student and even earlier, A level or FSC, it should be interdisciplinary. There should be more and more emphasis on interdisciplinarity. Gone are the days when people like me ran away from math and they found refuge in biology. And the ones who hated biology, they found refuge in mathematics. If you want to stay in science, then if you want to stay in science and then you want to achieve something big, you want to leave an impact in life, forget about Nobel. You don't do science for Nobel. No, none of us do science for Nobel. We are passionate about a subject and we are just working on finding the truth. So if you want to leave an impact in the field of your interest, if you can analyze data using MATLAB, or you can use R, you can use power of computing, you're an experimental biologist, and your math is not outstanding, it's not excellent, it's not very good, but good enough, you are a successful scientist, a very successful scientist, I tell you. And the bigger problems in the universe, as I said earlier, this century belongs to biology. This century is of life sciences, which is chemistry is life sciences, biology is life sciences, computational biology is life sciences. What you are doing in Corona, if you have followed earlier part, Ferguson from Imperial College, he is a theoretical physicist, moved into biology many years ago. He was using computational simulations and modelings, and his paper was cited, although later on he had to face the music because what he was predicting, it did not turn out to be true, but everybody relied on his predictions. Government of Pakistan, Americans, everybody want to analyze the data, but you need to know the biology before you can do something. Anyway, let's move to our today's lecture, and it's a very interesting lecture, I tell you, today. It's It's one of my favorite topics in biology. Not only mine, many molecular biologists you will talk to, I think more than 70% will, even if they are not working in this subject, they will name transposable elements as a fascinating topic. Transposable elements are basically also known as jumping genes or moving genes or mobile elements, selfish genes. All these four genes, actually, four names are synonymous for transposable elements. They were discovered in 1930s in Emerson's lab. This is Professor Emerson, together with Barbara McClintock and Beadle. Barbara and Beadle, they ended up winning Nobel. This picture is of 1929. Barbara won Nobel in 1986. Okay, I think she should have been awarded this Nobel much earlier. It was unfair, and she was lucky enough to be alive still in 1986. What are transposable elements? Why we call them jumping genes or moving genes or mobile elements? All this indicates, and even the transposition, transpose, it indicates that you have, for example, an element, a gene here, and on this chromosome, let's say this is chromosome 1, and this hops, this jumps to a totally new site on, let's say, chromosome 11 in humans, or wherever, whichever organism you talk about. So from their property of jumping from one place to another within the genome, they were referred to as jumping genes or moving genes or mobile elements. And more technically, they are called transposons. Transposons or transposable elements. Remember, in molecular biology, whenever there will be a word elements, it means we are talking something which is DNA based. I repeat again, whenever we used while talking in molecular biology or genetics or cell biology, whenever we are using about elements, word element is being used, it means you are referring to something in DNA. And factors, if we use factors, they are used to refer to proteins in cell. Okay, so they discovered transposable elements in Emerson lab in 1930s, and how they discovered it was a simple observation under microscope that so maize chromosomes are the corn, which you also know as. So there are 10 chromosomes, total 10 chromosomes in maize. The largest one is, so when you karyotype means you identify different sets of chromosomes in an organism. And mostly morphology is used. So maize karyotype, so the identification from 1 to 10 is that, chromosome 10 is the smallest and chromosome 1 is the largest. And what they observed that chromosome 9 was actually frequently broken at a particular locus. So chromosome 9 had a knob, you can see here. Both the homologs, they have knob, and it was frequently observed that at a particular locus, one of the chromosome is broken. As geneticists, they try to find out what is the reason, why they have why these breaks are there. And Barbara McClintock in Emerson's lab, they narrowed it down to two factors. They call them AC and DS. AC for activator, and DS is dissociator. These are just, how do they find? They will cross, let's say, a line which contains AC, cross AC with a line which contains DS. In the F1, you will have AC and DS together. If you find breakpoints are there, you will say, and if you have a line in which DS alone is there or AC alone is there, you don't see in these two lines these breaks, you will say, okay, maybe AC and DS have to be together. And since DS alone does not do not show such breakage, it maybe is due to activator. AC factor is required.
Sir?
Yeah.
Sir, what do you mean by activators and dissociators? Can you please explain that? I don't understand.
Yeah, soon. Just bear with me. We'll soon learn about these elements. Okay. In
Okay,
next
sir.
few slides. On this slide, what you should know is that there is break on chromosome 9 frequently. Okay. And they could narrow it down that there are two factors, AC and DS, in the maze, which are associated with this breakpoint. Okay, this is the broken chromosome, which is now useless for the system. So they did these beautiful experiments, and once they knew that chromosome 9 is involved, they chose certain mutations or phenotypic markers. For example, C is the gene. When it is homozygous recessive, it will be colorless. This is colorless. This part is colorless. Okay. And wild type C gives you pigmented. This is pigmented. Now, another gene on chromosome 9 is which gives you plump phenotype. And if you have homozygous recessive, the phenotype is shrunken. That's why SH. And VEXI is the gene. When it is wild type, it gives you shiny look of the kernel. When it is homozygous recessive, it's not shiny. So 3 phenotypic markers: colorless, shrunken, VEXI. However, if you have wild type, so it's a heterozygous, this will be normal wild type. I apologize for having a cup of coffee with me. Actually, I'm still suffering from the broken hand problem of this left hand, and my doctor has advised me. I usually don't drink coffee. My department will tell you. So they have recommended me to drink, I don't know, 6 cups of coffee a day, maybe. So it's a medicine, you can say. So when they observed that if the AC and the DS factors are present together, so here you can see the DS is there. If in the same plant you have AC as well, now the hypothesis was that AC acts on the DS element and breaks away the chromosome. It's called acentric. Acentric means without any centromere. It's a useless segment of chromosome. It's lost. Now, what will happen if such a breakpoint takes place? Now, they will behave as what?
Dominant. They will express their phenotype.
Recessive.
They will express the recessive phenotype because dominant alleles are gone. And if this is happening while the grains are growing on the corn cob, all this is what? This is result of mitosis हो रहा होगा ना, ठीक है? When this breakpoint happens, you will see the cells where breakpoint happened, its progeny shows colorless, shrunken, and not shiny phenotype.
Sir, when you say that the acentric fragment is lost, is it like destroyed?
Absolutely.
Okay.
So it is recognized as non-essential, as a what we call the within cellular nucleus and within cellular machinery. It recognized as a dangerous thing, and they destroy it and get rid of this. So this was due to chromosome breakage. Okay. They also Emerson lab also found a line in which, you know, they had the normal wild type gene for colorless, which is pigmented. This is mutated by presence of DS. So let's say you have wild type gene of pigmented, this gene, it is disrupted by the presence of, so DS is present here. By presence of DS here, what has happened? It has caused a mutation, disruption of the open reading frame, and it's muted. So we write it like small c DS. It means DS is present within, and due to presence of DS, you have a mutation. What they discovered that, and how they arrived to this allele, this was later, they had started a growing colorless corn, which should have been this yellow color or no color at all as compared to this one. And they discovered that there are patches of wild type color coming in. This was a surprise. And when they cloned this pigmented gene, they discovered there is a DS element sitting there. And if DS element is sitting there in colorless background, why we have these wild type colors then. Is it clear what I'm trying to explain?
Sir, I'm a little confused.
So they had a corn variety which was colorless. Okay.
Yeah.
But wild type for this and wild type for this, which means these two phenotypes were not there. When they grew colorless, they found such spots which are basically wild type, which is this one, which means we have in these spots, in these cells, we have wild type this gene. But we knew the seed we took and from where we grew this, that was a colorless mutant. When they cloned and discovered that the colorless gene is actually disrupted by DS element inside this. This is what I explained.
Okay, sir.
Now, they wanted to answer why we have then the pigmentation. And what they discovered that in these patches where we have wild type gene restored is actually due to jumping or transposition of DS element out of this locus. So DS element, that's why we write it C-DS, and it's an unstable allele of colorless. Unstable because DS can jump anytime when it will have AC activator together with it. So
Okay, sir.
AC helps DS to jump out. So this DS is no more there, and the wild type C gene is restored. No more disruption of the open reading frame. Clear?
Sir, I have a
Yes.
question.
Yeah.
So basically, DS is just a sequence of nucleotides. So how does that just jump out? Like it's removed by something?
Yeah. On next slide. Very good question.
Okay.
So-
Sir, I have a question.
Yeah, please ask question. Go ahead.
Sir, is it possible कि जो DS factor है, it comes on both the chromosomes in a homologous pair,
It can as well.
and that'll make the cell unviable, right? Because फिर वो दोनों उसकी side से genes जो हैं, they're gonna be removed.
When it jumps away, you never know. Maybe only one of the two DS elements they jump away.
Okay.
So on next slide, let's see. It's a, as I said, it's a strange but very fascinating topic. Now, what is actually happening? You have wild type colorless gene. It got disrupted by DS element, and this is referred to as C mutable DS. Okay, C mutable DS, which means C is disrupted by DS element. So the phenotype is normally, if this is wild type, you have pigmented kernel, uniform pigmented. If C got mutated, C mutable DS, this is the phenotype. However, if this parent gets access by crossing with another line where you bring in the AC element, now the DS moves out. Moving out of DS requires functional AC, and the result is when DS moves out, this gene is restored as wild type, and you see pigmented kernel. But what should be noticed and you should pay attention there. Why I do not have uniform pigmented? Because this event of jumping out or transposition of DS, it's not 100% of the cells. It's not taking place in 100% of the cells. So few the cells where, and you can then see difference in the size of the spot as well. Here, for example, this is a much smaller pigmented than this one. So it matters when this transposition took place in the life of these cells. If this transposition was early, it means they are progeny of, let's say this cell is the one where transposition took place. Now mitosis took place. All of them will show pigmented, and all of them are in this spot. More bigger the spot, it means the transposition event took place early during this development. And smaller the dot means it's later in development, like few cells ago, maybe this transposition has taken place, okay? However, another surprise came when they saw that, AC can also jump and it can also create C mutable AC. It can also give you such a colorless seed, but without having DS, AC can move out and give you this pigmented kernel. They called AC as autonomous and DS as non-autonomous elements. DS is non-autonomous because it cannot jump by itself. And AC is autonomous because it does not rely on anyone else. It can jump by itself. So it means DS interacts with AC, and we call this activator and dissociator system in maze. Now, what is an AC or a DS? So they suggested that AC is actually an allele of AC that has got mutated and converted into DS. Or DS is actually incomplete version of AC. Now, what is at molecular level AC and DS? What is present in AC and DS? So in AC as activator, there is an enzyme called transposase. Transposase is the enzyme which recognize very, very specific sequences at the border of the DS element. Right here, at the border of the DS element. And this transposase then, like a scissor, cuts here and there, and DS jumps and moves to a different chromosome, or maybe same chromosome, but a different position. So now you got it, why DS relies on the activator, because activator contains the gene, enzyme. So if you look at the DNA sequence of activator, it will also have these specific borders, but within AC element, you have an open reading frame which encodes this enzyme, which is called transposase, and this transposase recognizes the borders of DS, cuts it like scissor, or recognize borders of AC itself, and precisely cut it and move it to different position. Is it clear so far?
Yes.
Isn't it an interesting topic? I never thought in my lifetime when I was of your age, and I used to work in the fields of agriculture in my BS in Agriculture University of Islamabad, we used to see such kernels on corn, not with such a high frequency. These are the frequency is too high, okay? But few, 1, 2, or few seeds, and I was always afraid that there may be some larva there, insect, क्योंकि उसके अंदर सुंडी sometimes चलती हुई नजर आती थी. But my introduction to molecular biology took another 6 years or so, I think, after my BS. 4 years later, my BS. And when I discovered this, I learned something new, that okay, there are something in the genome. But still, before I could grasp the concept of transposon, it was another 3-4 years. And when I realized that there is something within the system which can cut itself at DNA level and hops itself to a different position within the same chromosome or a different chromosome. That was simply, I could not comprehend. I remember, for me, it was unbelievable that something like this can happen in a genome. Why we also call this selfish elements? I forgot to tell you. Selfish elements, because they say whenever it hops, let's say if it hops a transposon and there is a gene there, a normal functional gene in a plant or bacteria. So this transposon controls this one. It inactivates this, and the promoter element for this gene, which actually should have worked on this one, it starts working on this one. And transposon is happy, it keeps jumping. That's why it's called selfish gene as well. It kills others and hijacks their machine. Okay.
Sir, I'm still confused about what C mutable DS and C mutable DS are. So
C mutable, yes.
Gene.
C mutable DS means that the DS is sitting within. So this is the wild type C gene. If DS jumped and it's incorporated here, the C has become C mutable DS, and it's an unstable because DS can jump out. Clear?
Yes, sir.
Same is the case for C mutable AC as well. If AC lands into the colorless gene, C becomes mutated, isn't it?
Yes, sir.
C will become non-functional.
But sir, if they jump out, won't it cause a break in the? That's what they basically do, right? When they jump out, they cause a break in the chromosome from there.
It can, and there are two things. If when a transposon jumps out, it can be an imprecise excision. Let's talk about this. Imprecise means the DS jumped out and the repair machinery, it repaired, but it's not 100% open reading frame of wild type OR. Some nucleotides from neighboring side are also lost. In this case, C will still remain mutated. And the other jump is precise excision. In case of precise excision, when DS or any transposon will move out of this locus, you will get precisely the normal open reading frame, which was before the insertion of transposable element. The third possibility is what you are saying is very drastic one, where you will see, the acentric fragments. Double strand break is not repaired. Did I answer your question?
Yes, sir. Thank you.
Sir, I have another question. Sir, what determines whether the excision is going to be precise or imprecise?
Very good question. Very good question. We cannot pinpoint, we cannot really precisely tell you, even after nearly 100 years of research, that why we have imprecise and why we have precise excision. In next few slides, you will get a partial answer that when transposon jumps out of the chromosome, it somehow there's a recombination involved. And the answer I'm giving you is what we know from bacterial genetics. There we see precise excision and imprecise excision. And imprecise excision is when recombination is taking place between some elements in transposon and some genomic locus. Means, let's say in this case, the colorless gene, some sequence of colorless. Now it jumps out, it took away colorless segment as well. Not necessarily only here. In the case of imprecise excision, maybe when it jumps all the way from here to here, everything is deleted. Big chromosomal aberration. And precise is, so mostly I would say in my experience when I try to do imprecise excision, and in flies we do imprecise excision, you will see in later part of the lecture. In fly genetics, we wish to get imprecise excision because we want to delete neighboring gene, which is normally not mutant, and we want to create a mutation in our gene of interest, which is not right next to transpose, which is not, which is next to transposon some nucleotide away. We want to create a lethal allele of gene of our interest. And my experience tells me that mostly, I can't recall by heart, but it's a huge number. We get precise excision. Imprecise excision events are relatively few. Should I move on? So soon you will see the phenomenon for imprecise and the precise excision. Okay. So yeah. So once they knew transposable elements in maize way back in 1930s or so, you know, people in bacteria, they started working on, they were also, I mean, bacterial genetics was quite advanced because it was a simple organism, small genome. What they observed that, you know, because for long time, people did not believe Barbara McClintock. They said, you know, it could be a maize-specific phenomenon. It could be a plant-specific phenomenon. It's not happening. Just like me, even in 20th century, for me, I mean, I can argue that I was naive. My knowledge about molecular biology was almost non-existent. But even people in 40s, 50s, 60s who were really, you know, expert, highly accomplished, they really did not buy in the transposition or transposon elements. But bacterial geneticists also discovered similar phenomena. Here you can see this is the phage, bacteriophage. So it's a virus. So bacteriophage lambda. So the virus which infects bacteria, just like viruses infect us, humans. There are bacteria for, there are phages, viruses for bacteria as well. Now, when they observe that phage, when it integrates into the genome, so normally there are two life cycles, lytic cycle, and the second is lysogenic cycle. Lytic is when bacteria is infected by phage. Phage injects its DNA, it replicates, okay? And if there comes a stage where there are so many viruses, you know, their coat protein, they burst open and kill this bacteria and move to next, infect next bacteria. In this case, which is called lytic cycle, phage DNA does not integrate in bacterial genome. The other cycle is lysogenic, and in case of lysogenic cycle, what happens, phage, it injects its DNA into bacteria, and eventually the phage DNA gets incorporated or integrated into bacterial genome, and we call this lysogenic cycle. For example, so this is a lysogenic cycle. So phage DNA is recombining. There is some kind of homology here on the phage DNA, viral DNA and bacterial DNA. And this leads to integration of phage DNA into E. coli chromosome. And there are markers, galactose gene of bacteria and biotin gene are neighboring phage DNA. Now, when this phage moves out of this bacteria, where it is quietly sitting, there are two possibilities. One is what we just talked about, precise excision, or what here it is written as normal outlooping. Normal outlooping means the sites which were used to recombine, there's going to be recombination again there. And you have, if this is the phage genome, marker 1, 2, and 3, this is the time when it came in. After precise excision or looping out, normal loop, we have perfectly normal phage DNA, just like when it entered bacteria. The bacterial markers, galactose and biotin, they are still there. However, in case of imprecise or abnormal looping, outlooping, now you see what is happening. The sites where recombination took place, it was not used to loop out or move out. Rather, so you have marker 1, marker 2, marker 1, marker 2, and recombination somehow took place at a different site within the phage genome. In such a case, after looping out, you can now see it has taken away the bacterial gene, galactose, together with it. And as a result of imprecise excision, the phage DNA is now marker 1, 2, and galactose gene of the bacteria. And it has left its third marker within the bacterial genome. This is normal, you know, observation in, I think, 50s and 60s, they were observing this. They did not know what is happening. They had no idea. You know, they said, okay, it could be due to recombination. You know, this is recombination is taking place here, no? But a little more careful analysis took them to discovery of transposons, which we'll see. Now, what they did, they said, now what we have, look, this this bacteria, which has lost galactose, isn't it now gal minus? Is it okay? Hello?
Yes, sir.
I think so, sir.
In this phage, which carries gal plus, can we write it like this? Lambda gal plus.
Yeah.
So this was, you know, a simple tool for bacterial geneticists. They used to use lambda gal plus to complement gal minus mutants to see if gal minus mutation is dominant or negative. So you're getting what they did from a simple observation of such normal life cycle and what is happening in nature with relationship of bacteria and phages. They developed some genetic tools for complementing bacteria, which we'll talk 3-4 lectures down. However, they also wanted to know what is happening here. Why? What is different between lambda gal plus and lambda gal minus? Virus, which is gal minus, which means this one, normal, and the one gal plus, what is special there? And how it is generated? So they simply ran what we call density centrifugation. And density centrifugation means, you know, you have a gradient of, let's say, sucrose solution, okay, based on the density. And what we'll have, let's say, let's write it down like 1 molar sucrose, then 800 millimolar, 700 millimolar, 600 millimolar. We have layers of, you know, sucrose, and then on top of that we overlay the viruses which we have isolated, which are galactose plus and galactose minus. We don't know what is different between them. We just know that here I have the galactose minus virus, and here I have galactose. I load them both in the same tube. I run then the centrifuge at a very, very high speed, like 40,000 or 80,000 RPM revolutions per minute. And then I see, based on the density, the viruses, I see 2 discrete rings on specific density. And when I analyze this, I observe that, you know, this one is the one which is gal plus virus is layering here, and the gal minus virus is layering there. So this is based on their bond density. This is based on their density. So gal minus sediments there and gal plus sediments there. Now, what it tells us about transposons, it doesn't tell us anything. But this led to discovery of what we call IS elements in bacteria, because what they observed that when phage outloops abnormally, actually it is recombining at specialized sequences within the genome, which are called insertion sequences. Insertion sequence. And these are the sequences which are also used by the phage to recombine something like this to begin with. So here, this is how you do these experiments. So you have E. coli bacteria, which is gal minus. You infect it with phage lambda, and if this phage lambda is E. coli gal minus strain,
Sir,
yeah,
so how did the previous experiment kind of show them that help them figure out the IS elements? I don't understand how does that
yeah.
correlate?
So
chemically?
what they discovered, because here now they can easily isolate DNA from gal plus. Clear?
Yeah, yeah.
Because it separate, all of them are gal plus viruses. You isolate their DNA, and once you know their sequences, you discover that these gal plus which outlooped within the genome, they actually carry more or less same sequences. So they are preferred hotspots where virus outloops by recombining. And it's not that virus likes galactose. It may recombine on this side as well and take away BIO plus gene if the hotspot is like this DNA element is there. And once they carefully analyze the DNA sequence, they name these sequences what we call insertion sequences. And there are, in bacteria, they have IS1, IS2, 3, 4, 5. So you can see now the total number of copies of IS1 elements, they're 5 to 8 on chromosome. And length of one IS element, IS1, is 763 base pair. And also there are 18 to 23 base pairs inverted repeat. So let's say this is IS1, 768 base pair, but on both sides you have inverted repeat. Inverted repeat means you have DNA, same DNA in inverted orientation. And such IS2, 3, 4, 5, they also characterize their sequences. Now these insertion elements are actually, they were behaving as transposons. I was going to explain this one to you, and I think I have some mistake, or let me carefully first look at this figure. So we have E. coli gal minus strain, which means E. coli lacks gal here. We infect it with phage. Okay, phage DNA comes in. Now you isolate all the phages from here, from this, okay, and from individual synthesized radioactively labeled RNA. From these ones we transcribe there. So viral genome is transcribed, make RNA, and hybridize with gal minus. Hybridize with gal minus. There's a mistake maybe. Hybridize with gal minus. Why gal minus? So we are starting with gal minus. If this viral genome, has integrated in This is already gal minus bacteria. The virus has to be gal plus. Sorry. So virus has to be gal plus. Okay. And the virus which we use to infect this lambda bacteriophage is, its genotype is gal plus. Bacteria is gal minus. If this gal plus recombines, okay, and now you have within bacteria, if recombination has taken place,
Sir, I think this experiment is to show that the gal minus gene from E. coli is being carried by the phage. So gal minus E. coli strain was infected with phage lambda, and then what we do, we synthesize, we take out the RNA and radioactively labeled, and then we will hybridize with the gal minus of that E. coli strain के वो उसके साथ hybridize कर रहा है या नहीं. So in order to find out के जो gene phage लेकर आ रहा है gal minus वाली वो E. coli ही की है. So in order to hybridize with that, I think that's the.
So why do we need gal minus strain then of bacteria? We can do it simply with gal plus, no? My worry is if I have had a typo here. I'll get back to you with the explanation for this. This is basically, so what Najma is saying, that's also true, and what I'm trying to figure out if this was gal plus or whatever. Anyway, whether gal plus or gal minus. So if, forget about this, this is a normal wild type phage. This is what Najma is telling us. So normal wild type phage infects gal minus, and if after outlooping of virus, if lambda has taken away gal minus, once we will transcribe viral genome, make its RNA, which is radioactively labeled. And on a northern blot, if we hybridize it with gal minus strain, we will know that phage has taken away bacterial gal minus locus. And this gives us an idea, and we can sequence the region within the phage which has enabled phage to take away gal minus or gal plus, which means the bacterial genome it has taken away.
Yeah, sir, I want to add one thing कि E. coli gal minus strain का तो मतलब है कि उसमें से gal gene निकल गया है.
Yeah.
So वो gal plus ही होगा कि हमने उसको normal phage से infect किया है और end पे हमने उसको gal plus के RNA के साथ hybridize किया है.
Yeah, that's what I meant, that मुझे यहाँ पे typo लग रहा था कि we had gal plus. So once phage will take it away, गैल माइनस का तो मतलब हुआ कि उसमें लोकस ही नहीं है, तो हमने हाइब्रिडाइज करके तो पता ही नहीं चलेगा वायरस लेके आया कि नहीं.
जी सर.
So once we have gal plus, if we will hybridize with gal plus gene, we will know that the lambda has taken away galactose gene. And they characterize that these sequences, which they named as insertion sequences, IS elements, they were involved in this outlooping of viruses. Outlooping as well as insertion of virus when virus enters into the bacterial genome. And then there are these characteristics of, I told you IS1 has repeats. Now these repeats are very important, 18 to 23 nuclear inverted repeats, all the ones they have it. Okay. Now, what are the characteristics of bacterial IS elements? Bacterial IS elements, as I told you, they do have inverted repeats. Let's say here and here we have 25 nucleotide, exact sequence, 100% sequence, except that it is inverted. And then within the inverted repeats, we have enzyme called transposase. Now this transposase is the enzyme, which when gets transcribed, it recognizes these repeats, and it, like a scissor, it cuts them from here and helps them jump to somewhere else. Another very interesting discovery while discovering the transposons in bacteria came as a surprise, that the antibiotic genes, drug resistance genes, they reside on these mobile genetic elements. So in bacteria, for example, there was a bacteria which is tetracycline resistant. Another bacteria which is, for example, gentamicin resistant. These are antibiotics. What they discovered that actually these genes, they reside on transposable elements. Here is an example. So transposons in bacteria are classified in two classes. One is composite transposon. Composite transposons are within two IS elements, you have tetracycline gene, an antibiotic resistance gene. They named it TN10, transposon 10. TN10, transposon 10. And so I repeat, between two identical IS elements, insertion sequences, there is gene for transposase. IS10 in inverted orientation, and in the middle you have tetracycline. The other one is simple transposon. Simple transposon means you have, few nucleotide inverted repeat, then ampicillin resistance gene, then there's transposase, there's a repressor element, DNA sequence which is recognized by repressors to keep them switched on. And simple transposons, they are simply flanked by 50 nucleotide inverted repeat. Here what you have IS10. What was IS10? If we go back, IS elements are, 700, let's say IS1 is 768, IS3 is 1400, and there is repeat here as well. So for example, in case of composite transposon, you will have repeat here, inverted repeat there, and then there will be an open reading frame somewhere there. So two identical IS elements, which is IS10, contain tetracycline within, which is the antibiotic resistance gene. And simple transposon, just 50 nucleotide inverted repeat, having transposase gene in the middle, as well as ampicillin resistance gene and antibiotic resistance gene, etc.
Sir, I have a question.
Please.
So basically these inverted regions are the borders that the transposase recognizes?
Yes.
Okay.
So how transposition takes place? None of you asked how this transpose is. It comes and cuts the DNA. So here is a simple phenomenon, for example, shown that you have, for example, you have this sequence, nearly 12 nucleotides or so. 11 are here, so 11 to 12 nucleotides. The transposase, it cuts DNA like this. So the sequence is AGG, TAA, GGTAG. There is no symmetry involved here. Okay, it cuts it like this, and then the transposase cut leads to staggered end, to sticky end. This is sticky to this one, no? But after double strand break, you have such two sticky ends. Once transposase cuts, double strand break takes place, the DNA is separated like this, and transposable element, it hops within this sequence. And host machinery is trying to repair the double strand break, it immediately repairs this one like this. You see now there is nearly 5 nucleotide long, which is exactly identical: CCTTA, CCTTA, GGAAT, GGAAT. It becomes duplicated on both sides of transposon. 5 pair direct repeat. It's direct. ATTCC, ATTCC, okay, or GGAAT, GGAAT. This is indication that transposon has landed. This is a basic characteristic of transposase mediated transposition of a transposable element. And there are 2 different modes of transposition. One is, for example, a replicative mode of transposition. Replicative is easy way of remembering is replication. What happens when transposon jumps from its native site to a new site? For example, you have A, B, C, D locus. Now it jumps to a new position between C and D. So it does not leave its original position. It remains there intact, but it also becomes integrated in a new locus, CD. So that's why it's replicative. And conservative is, for example, TN10 transposon, when it jumps from this locus between C and D, it loses its original position, A and B. So it leaves position between A and B and integrates into the new landing site. We call this conservative mode of transposition. Is it clear till here?
Sir, I have a question.
G.
Sir, these transposons, when they're cut out of the DNA segment, are they reutilized or are they just destroyed?
Can you repeat your question, please?
Sir, when they're cut off, right, cut out of the DNA segment, are they reutilized or are they like just destroyed?
Yeah, so we should always pray that Allah Mia, may this transposition never happens, because they are not destroyed. If they hops, if they jumps, you know, that is havoc to genome. A large scale genome instability is the answer. Large scale mutations. It will not just jump and occupy this position. It may go to another position, another position, another position, another position. And the more it jumps, the bigger number of genes it mutates, and our chances of getting cancer or death, whatever, is very high. Normally, transposons, they are under very tight control. Very tight control means they are silent. Their transposase gene is switched off. Never, ever it is going to switch on generation after generation in a species life. If they jump, they are going to kill you. Yeah.
What motivates the, what is the reason that the transposon actually decides to jump from one gene to another? There must be some sort of condition or some factor that causes this.
Very good question. So normally they jump under stress. For example, in plants, they have seen if plants are under prolonged stress, there is threat to their survival, then transposons start hopping.
Is this in an attempt to make the plant more genetically favorable?
Yeah, they are trying to adapt.
Okay. Okay.
So
Also,
transposable.
sir,
Yeah, please.
sir, are they present in all organisms?
They are
Silent or
present
not?
in all organisms. In humans, there are millions of copies. We are going
Sir,
to, as we move on. Yeah.
sir, when you say that the organism is under stress, what do you like? Stress is in like they can't survive or something like that?
Let's say prolonged drought stress, heat stress, when there is life and death danger. Now, an obvious question you should have asked is, what is mechanism of keeping them off? Keeping transposons under control? Or how transposons are kept silent or kept immobile? So these are the epigenetic phenomena. And one of the epigenetic phenomenon is, for example, DNA methylation. Wherever you will have transposon, this region will be highly methylated in plants and in humans, okay, by enzymes called DNA methyl transferases. And when you have high methylation, there is no transcription. Transposase gene will not be transcribed. And when transposase is not transcribed, there's nothing available to cut it and help it move out. Now, once you're under tremendous stress, idea is that, yeah, now cellular machinery is these DNA methyltransferases, everything is getting compromised, and transposase, they get an opportunity to start jumping. Did I answer your questions?
Yes, sir. Thank you.
Okay. Now, in bacteria, there's also, as I told you, they discovered that the way bacteria become drug resistant is, the intelligent machinery of bacteria, they uses transposons as a tool, and they can exchange genetic material. Although bacteria do not undergo sexual reproduction like males and females, but bacteria can exchange genetic material. They come next to each other, and there's a specialized structure called pillars, which connects two bacteria of specific genotypes, and then one acts as donor. It gives its genetic material to other, and mostly these are plasmids, and we'll talk about some of them. For example, in bacteria, one of the replicative mode of transposition is, for example, here is shown TN3, this one. This is a plasmid, donor plasmid, on which TN3. So they have, based on the, for example, here name is given TN10. TN10 is, number is, it's a indication it carries tetracycline. Similarly, if you have TN5, TN3, I think TN3 is another antibiotic. So based on the antibiotic resistance gene, these numbers are given. This is to my little knowledge. So TN3 encoded transposase is being given by a donor, and this is the target plasmid of the other bacteria, or within the same bacteria, because same bacteria may contain many plasmids. So this is its genome, and then there are plasmids within the bacteria. It has acquired TN3 plasmid from another bacteria. Now, once it comes in, it tries to incorporate donor TN3, because TN3 is, for example, kanamycin. There is a recombination, very specialized recombination that is mediated by transposase. So it creates cuts here, this arrow and this arrow. It leads to single-stranded cuts there and there. And transposes then links DNA of the donor sequence with DNA, because recipient also plasmid gets single strand cuts. So there is ligation here, and then rest is simply repaired. You will start having its polymerization. We call such a structure when two plasmids are integrated into each other, we call such a plasmid co-integrate. And once it's done, you resolve the co-integrate simply again by nicking. And what you have now, you had the whole TN3 from donor is incorporated into recipient plasmid. Host plasmid also retains TN3, but the new plasmid also has TN3. This is how bacteria, let's say, you may have heard about multiple drug resistance is a big problem. Bacteria are becoming more and more and more resistant against antibiotics, and it's a big problem in medicine. The reason is we give antibiotics indiscriminately when there's no need of antibiotics. We are giving patients antibiotics. These bacteria are, and sometimes if antibiotics are suboptimal doses, if antibiotic quality is not good, this is a reason bacteria is going to become resistant. Now, these bacteria do not go anywhere through our feces, through when we cough. It depends what kind of bacteria we are talking about. They live in the environment. When these bacteria will come closer to another bacteria, let's say one is TN3, the other is TN10, they exchange their material and both become, let's say, one become TN10 plus TN3 as well, tetracycline plus kanamycin resistant. And this is due to jumping of these transposons across their population. Now, let's look at the eukaryotic transposable element. All that was up to the prokaryotic transposons. We started off with eukaryotic transposons, which were ACDS element of maize. Then we went to bacteria because, people were not willing to buy that transposons are a reality in the genome. And bacterial geneticists, they showed repeatedly that, these IS elements and these TN elements, TN10, TN3, etc., these are actually indeed transposable elements. In eukaryotes, there are two major classes of transposable elements. One are DNA-based or DNA transposons. What DNA transposons do, they simply either copy or they excise based on, you remember the replicative mode of transposition or conservative transposition. And copy of DNA, they leave their original place or they can retain their original place. They jump to a target sequence, new position on chromosomes. And the other class, which is a very specialized class, is called retroelements, or also they are called retrotransposons. What retrotransposons are doing? This is a very dangerous class. They don't need to leave their position. They stay where they are. They simply get transcribed in an RNA. These transposons, which are called retrotransposons, they also have an enzyme which is called reverse transcriptase. You remember, for transcription of normal genes in our system, we have RNA polymerase. And what RNA polymerase does, it transcribes DNA into RNA. These intelligent elements, they have specialized polymerase, which we call reverse transcriptase, which takes up their RNA and makes DNA out of this, reverse transcribes this. Okay, it makes a copy of this DNA. And now this product of reverse transcription is free to go anywhere in the genome. This is very dangerous situation. Why? Here we have one copy of DNA, excises itself, lands, job is done. One copy, let's say it left its original position, jumped to new position. And if you are lucky, let's say it jumped into an intergenic region, no essential gene is there, nothing, no danger zone, and you're still safe. Here, same RNA can be transcribed multiple times through reverse transcription, and many products of reverse transcription will be made. Now, each one of them can jump to independent site, and we are screwed up. There are millions of ALU sequences, and these are, the transposable elements, ALU. And this insertion is also permanent because this is not going to leave its position. Once it lands, it just transcribes, and the product of reverse transcription is then going to hop around.
Sir?
Yeah.
So why can't it leave its position?
Why it cannot leave its position? Because it does not have a transposase like DNA transposase, which recognize its sites and then moves it. It relies on an RNA intermediate.
Okay, sir.
I explain you the word retrotransposons or retroviruses, you may have heard. Retroviruses. HIV virus, human immunodeficiency syndrome virus, is actually a retrovirus. It has the same phenomenon. For example, this is the genome of a retrovirus. It is bound by reverse transcriptase. When virus infects a living cell, now the reverse transcriptase is going to make copies. Through reverse transcription. Now DNA copy is made. This DNA copy then goes and jumps into the host genome. When viral genome is integrated into the host genome, we call this provirus. Now, this provirus can now synthesize it gets transcribed. Messenger RNAs, viral mRNA is made, which can be packaged like new virus, or it can be simply through process of reverse transcription. It can again get reverse transcribed and jump on different other positions of the same cell. But when it infects more cells, of course, once it lands in, it makes its RNA, which gets incorporated into the viral capsid. And this phage, this retrovirus, once it will leave this cell, they will infect all the neighboring cells through bloodstream or whatever. Single virus has played havoc. Now, this is what we just learned about the class 1 retrotransposon. This is happening in retroviruses as well. So the one thing which is common between retroviruses and retrotransposition is RNA intermediate, reverse transcription. Okay, how and what kind of retroviruses or what kind of retrotransposons are known? Gary Funk's lab, they discovered TY1 in the yeast. Now, what are the characteristics of retrovirus? You have LTR. LTR means long terminal repeats. What is this? Direct repeat. Same 100% same nucleus, 1 kb in direct repeat. Now, this is a retrovirus. MMLV is a virus. Look at its genome organization: LTR, GAG gene, polymerase gene, envelope gene. Envelope is the membranous capsid within which this genome is packaged together with reverse transcriptase. Now, if we look at the retrotransposons, look at the retrotransposons. These are retrotransposons. Dangerously similar genome. LTR, LTR, GAG, pol, only the envelope is missing. Look at the fly, copia gene. LTR, LTR, GAG, pol. L1 in humans, lines. Open reading frame, pol, poly A, tail. There are no LTRs there. Now, Gary Funk's lab did a very intelligent experiment to prove, and you have to design such an experiment when you are working with transposons, retrotransposons in particular, and you discover a new retrotransposon, to prove that TY1 is a retrotransposon. Now, how TY1 transposes via RNA intermediate, this was the discovery that TY1 is present in the DNA, and once it is in the DNA, you see there are 5 base paired direct repeat. This is what we learned when transposon lands into the genome. There's 5 nucleotide direct repeat. This shows, yeah, this is a transposon which has landed here. Now there's a messenger RNA being transcribed from this locus. This messenger RNA goes to the nucleus. Reverse transcriptase is made from this messenger RNA. This reverse transcriptase then makes copies of this messenger RNA, and reverse transcription leads to synthesis of complete retrotransposon, which is this one. This product of reverse transcription now goes somewhere else in the genome. Somewhere else in the genome. Now, how do we prove that it involved reverse transcription or RNA intermediate? And it simply could have, because this copy is 100% similar to this one. So it could have simply jumped like DNA transposon. How do we prove there's an RNA intermediate? So they designed a very intelligent experiment, and that was they cloned TY element on a plasmid. You see LTI, and then the coding region. They cloned the whole transposon under galactose inducible promoter. So in the yeast cell, this transposon messenger RNA will only be produced when in the media we will have galactose. So this is inducible system. In the absence of galactose, it will not transcribe. Another very important and intelligent thing they did, they inserted an intron. They inserted an intron within the open reading frame of TY1 coding region. Put everything, this plasmid, into the yeast and grew yeast in the presence of galactose. Adding galactose must have resulted in transcription, which is the messenger RNA synthesis. If it gets reverse transcribed, before reverse transcription, since there is an intron and yeast is a eukaryote, it splices out the intron. And if we see new sites of transposition in bacterial chromosomes, now TY1 transposed to new sites in the genome. If the new sites are without an intron, this is a proof I went through RNA intermediate. This was a very intelligent experiment by the Gary Funk's lab. Is it clear till now?
Yes.
Okay, I think this is the last slide. We can stop it here. Yeah, we better stop it here. Okay. So because this is I would like to spend more time on this flies, and this requires, you know, more concentration, and it's already time. So by now what we have learned, we have seen, we started transposons in maize, which was AC and DS elements. We went to bacterial transposons, which were, you know, TN5, TN10, etc., IS elements. And we have seen retrotransposons in eukaryotes. And we saw examples of TY1 in yeast. And also we have talked about DNA transposons, which do not require any RNA intermediate to jump around. So in next lecture, we'll talk about DNA transposons still in eukaryotes, but you know, in flies and in humans again. Okay, any questions? Any questions? No questions? Okay, thank you very much then. I wish you.
So we had to announce the quiz.
Yeah, so there will be quiz on next Tuesday. Next Tuesday, yeah, na?
The coming Tuesday.
Yeah, today is a Tuesday, so coming Tuesday next year.
Right.
So there will
Yes,
be
sir.
a quiz next Tuesday, ladies and gentlemen. And the quiz will be from lectures which we have gone through after the classical genetics, which included DNA mutations, chromosomal point mutations, etc., chromosomal aberrations, and transposons. Okay?
So how long
During
will
class
it?
time?
It will be short, don't worry. So these 3 lectures which we have covered so far. Clear?
Yes.
Yes, sir.
Yes, not so happy. Huh? Chale, thank you very much. Allah Hafiz.
Thank you, sir. Allah Hafiz.
Office.